Hello everyone, this video is about polysaccharides. This is a part 2 of polysaccharides. In the previous video also we have discussed about the polysaccharides. In that video we have only discussed in detail about starch and glycogen. In today's video we'll be taking up cellulose and chitin in detail. But before that, let me give you a quick review about that. We have already studied about carbohydrates, and in carbohydrates we have studied that these can either be monosaccharides, or these can be oligosaccharides, or these can be polysaccharides. Monosaccharides will be made up of only one type of saccharide or sugar. Oligosaccharide will be made up of 2 to 10 units of saccharides or sugar. Polysaccharides are made up of more than 10 units of saccharides or sugar. Now, in polysaccharides, we also studied that these can either be homopolysaccharides or these can be heteropolysaccharides. Now, what are homopolysaccharides? Homopolysaccharides are the saccharides that are made up of a single unit of sugar. For example, glucose, 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 and so on. So it will be a homopolysaccharide. Heteropolysaccharides are the saccharides or sugars that are made up of, are those kinds of carbohydrates that are made up of different kinds of sugars. It can either be glucose, galactose, glucose and galactose, and glucose and galactose, and so on. So these are the polysaccharides. In the previous video on polysaccharides, we have already studied about starch. In starch, we have taken up amylose, and we have studied the structure of amylopectin as well, amylopectin. And in the previous video, we also studied about glycogen. In today's video, we'll be taking up cellulose and chitin. So first of all, let's start with cellulose. First of all, we will have cellulose. Now, cellulose is made up of repetitive units of beta glucose. Now, what is the structure of beta glucose? Let me explain that. As we know that glucose is a 6-carbon structure. We have an oxygen. We have carbon number 1, carbon 2, carbon 3, carbon 4, and here we have carbon 5 and carbon 6. Now, here we'll have OH, 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 OH. So, this is the structure of a beta glucose. Beta glucose. Now, when the repetitive units of beta glucose will join together in an inverted manner. Inverted manner means here we have started, this is carbon 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6. In an alternate way, in an inverted way, this will look something like this. We'll have an oxygen, we'll have CH2OH, means the numbering will start like this: carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and carbon number 6. So in this way, an upright, inverted, then again we'll have an upright structure which will have oxygen here, and then we'll again have an inverted structure like this. In this way, when these join together, these will form a linear chain of cellulose. Now, first of all, let's discuss some properties of cellulose. This is the most abundant carbohydrate on the earth, which is found on earth. It is a homopolysaccharide. We have already studied it's a homopolysaccharide because it is made up of only beta glucose units in upright and inverted manner. It is formed by the condensation of beta glucoses, and it is the major structural carbohydrate. Structural carbohydrate of plants means the structure of plants is made up of cellulose. Example, cotton and paper. These are the purest forms of cellulose. Now, let me show you the structure for this. Here, can you see? This is our beta glucose. This is the beta glucose. Again beta glucose. Again beta glucose. and so on. This is an upright one. This is inverted, sorry, this one is the inverted glucose. This is upright. In inverted glucose, the numbering will start from here. 1, 2, 3, 4, 5, and 6. Whereas in this one, the upright one, the numbering starts like this. 1, 2, 3, 4, 5, and 6. Now, we can see that the bond, the loss of water, takes place from carbon number 1 of this glucose and carbon number 4 of this glucose. So that is why this linkage is called as beta 1,4 glycosidic linkage. So basically, cellulose is a structure that is formed of repetitive units of beta glucose, and these beta glucose are joined together by beta 1,4 glycosidic linkages in upright and inverted manner. Now again, this one is the upright structure. This is the inverted structure because oxygen is here. Again, we'll start numbering it. Carbon 1, 2, 3, 4, 5, and 6. Now again, you can see that the carbon, that the water is lost from carbon number 1 of this glucose structure and carbon number 4. So again, this bond will be beta 1,4 glycosidic linkage. In this way, different linkages will take place. These different beta 1,4 glycosidic linkages will take place, and a continuous chain of cellulose will be formed. Now, let me show you a picture for this, a better picture for this. So here we have, here you can see one beta glucose, another beta glucose, another beta glucose. This is a linear chain. This is an unbranched structure, and you can see in between carbon number 1 and 4 there is beta 1,4 glycosidic linkage. I hope this is clear with everyone. Now, how does this beta glucose look in the plants? Let's see. So here we have this, as we have studied that the cellulose, it is the most abundant material found in the plants. This is the basic structural material of a plant. So here we have the plant cell. In the plant cell wall, especially the wall, we have these cellulose fibers. These cellulose fibers are divided into larger macrofibrils, and macrofibrils are made up of microfibril. This microfibril, this single thread, you can see it is the cellulose molecule. Now see, this is one glucose, beta glucose, another beta glucose, another beta glucose, and in between them you have beta 1,4 carbon linkage. So I hope the structure of cellulose is clear with everyone. Now let's start with the next structure, which is the chitin. Now chitin is the structural material. It forms the cell wall of fungi. That is why this is also called as the fungal cellulose. This is the second most abundant organic molecule that is found on the earth, and it is a structural carbohydrate of cell walls of fungus that we have already discussed, and it also forms the exoskeleton of arthropods. Exoskeleton means the external, exo means the external skeleton of arthropods. What are arthropods? Arthro means joined, and poda means legs. So those organisms that have joined legs, which we also classify as insects. So the cell wall of all the insects is made up of chitin. This chitin is actually a repetitive unit of N-acetyl glucosamine. So when different units of N-acetyl glucosamine will combine together, it will form an elongated chain of chitin. Now, what is N-acetyl glucosamine? N-acetyl glucosamine is also a derivative of glucose only. See, this is the structure of glucose that we have already studied. Here. Now, what happens is that This is the structure of glucose. Now, what happens in glucosamine is that this OH is replaced by NH2. In glucosamine, this OH structure is replaced by NH2. So this is basically the structure of glucosamine. Now, what is N-acetyl glucosamine? In N-acetyl glucosamine, one H is replaced by COCH3 group. Now this part is called as glucosamine, and when COCH3 group attaches to this part, this is called as N-acetyl glucosamine. So chitin is formed by repetitive units of N-acetyl glucosamine. Now another thing which is very important here is that N-acetyl glucosamine will be again joined in inverted and upright manner. Means in one structure, NHCOCH3 means this acetyl group, this glucosamine group will be joined on the lower part, and in another structure it will be joined on the upper part. Let me show you how does that look. It looks something like this. See, this is the first structure of an acetyl glucosamine. In this structure, as you can see, we have this acetyl glucosamine group here. So this is the upright one. This is the inverted one. Now we have upright and we have inverted. But if we see this complete structure, this is N-acetyl glucosamine 1. This complete is N-acetyl glucosamine 2. This is third structure, fourth structure, and so on. This will continue. Again, this chitin is an unbranched chain. Now, what happens is that when we start numbering it again, see, this is carbon 1, 2, 3, 4, 5, and 6. In this one, we have carbon number, this is carbon number 1, 2, 3, 4, 5, and 6. So now you can see that the water molecule is lost between carbon number 1 of this N-acetyl glucosamine and carbon number 4 of here. So again, this will be a beta 1,4 glycosidic linkage. This is also a 1,4 glycosidic linkage. How? This is carbon 1, 2, 3, 4, 5, and 6. So carbon, now again the dehydration reaction or this condensation takes place between carbon number 1 and carbon number 4. So this bond is again our beta 1,4 glycosidic linkage. So in this way the chain will continue and we'll get a structure of chitin. I hope this structure of chitin and cellulose is clear with everyone. If there is any more detailed structure that you want to study about, you can always write to us. Your feedback is most welcome. Thank you for watching the video.